Okay, today we're going to be starting the new unit, unit number 3, which includes the urinary system, reproductive system, skeletal muscle system, as well as the endocrine system. So to start off the unit, we will begin with the urinary system, which actually it's quite a lot of information. Actually, everything in the third unit is quite challenging compared to the earlier stuff that you've learned. So we're in the final stretch of the course, so it only makes sense that things get a little bit progressively harder, right, as the course goes on. So here we go. First question: list 3 functions of the urinary system. So the first function of the urinary system is the ability to perform excretion. Okay. So excretion is basically the ability to remove waste from blood. Okay. And then that in turn would produce urine. Okay. The second function of the urinary system is elimination. Okay. Elimination is the ability to remove waste from the body. Okay, so the difference is, after you remove the waste from the blood, it's still in the body, and then elimination is the process of actually peeing it out, right? Getting it literally out of your body. And finally, the last function that we talked about in the urinary system is the ability to help maintain homeostasis. Yeah, so specifically in the lecture, we talk about the ability to maintain water salt balance. Okay. The ability to maintain blood pressure. And one more is the ability to buffer the blood. Okay, so there are more examples of how the urinary system helps maintain homeostasis, but these are the ones that was covered in the lecture. So next, we're going to list the part of a nephron. Now, in case you've forgotten, the nephron is the structural and functional unit. We say functional unit of the kidney. Okay, and it looks like a crazy straw, right? A straw that has been all twisted up, and there are different parts to it. Okay, so let me just quickly show you. So here is a diagram of a nephron, and this is labeled in the lecture recordings. I know there's only 6 people watch the recordings. I mean, you guys are here for the tutorial and on time, so I assume you guys have watched it. But for those of you who have not, it's very important that you do, because there's no way to just learn this on your own, by reading the PowerPoints or something like that. Okay, so the nephron has different parts to it, and we can just draw one here, as we're labeling it. So there is a ball at the beginning, okay, and then it's gonna go down. It's gonna come back up. And then it's going to go down again. Okay, so typically, part of it will be in the renal cortex, which is the outer layer of the kidney, and then you would have a loop that goes down into the renal medulla. Okay, so starting at the Bowman's capsule, that's what this thing is, Bowman's capsule. We're going to move on to the proximal tubule. Okay, proximal tubule. And excuse me, and we're gonna have the descending limb, followed by the ascending limb. Okay, and together, the descending limb and the ascending limb forms. Let me just quickly check to see if you guys know what I'm talking about. Together, what do we call them? Anybody know? What is this U-shaped structure? What is it called? Does anybody know? Okay, so this is the loop of Henle. All right. Loop of Henle. I'm just gonna put LOH here. And over here we have the distal tubular. Okay. Distal tubular. And finally, we have the collecting duct. Okay. So the content inside, okay, the stuff that's inside the nephron, the fluid, the liquid that's inside the nephron, okay, that is what we call the filtrate. Okay, and the filtrate is what will become the urine. And filtrate is what will become the urine. Okay, list all the structure a drop of urine will pass through after exiting the collecting duct. So from here, where does it go, right? Okay, so maybe just for the sake of reviewing, let's draw a kidney. We'll quickly talk about the structure. Okay, you should draw this with me, especially if you did not watch the recordings. Yeah, you should draw this with me so you know where things are. First of all, we have two kidneys, right? So there's a kidney, there's a kidney. Okay, kidney. Okay. On top of the kidney, we have a gland. Can someone tell me what this gland is? What is the name of that gland that sits on top of the kidney? Anyone at all? What do we call that?
Adrenal gland.
That is the adrenal gland. Very good, Shamira. Adrenal gland. Okay. So the adrenal gland, it has its own function, but one of the things that it does is it actually secretes a hormone that it's related to the, kidney function that we'll talk about later. So you have your abdominal aorta coming down from the heart. Then you're going to have your renal artery. Okay. And of course, you're going to have your renal veins as well, coming back out like this. Okay, so this is going to be going to the inferior vena cava, and then this is going to be coming down from the aorta. Okay, so we call these a renal artery. And the blue one is a renal vein. Okay, we're looking at the front. So this is the left kidney, this is the right kidney. Okay. I'll give you a moment to catch up. Any questions so far? All right. So from the kidney, you're going to have two things that are coming down. These are tubes. Those are your ureters. Okay, you have two ureters and they connect the kidney to this thing right here. Anyone know what that is? Help me out here. What is that? Anybody can tell me what that is?
Urinary bladder, maybe?
That is urinary bladder. Very good. Urinary bladder. Okay, so it's gonna fill up with urine and it's gonna come out from this thing. That is the urethra. Okay, and then the pee is gonna come out. Okay, so that's the ureters. You have two and one urethra. Okay, so urethra is longer in male than female, right, because of the anatomy. And that's basically the, key structures involved in the urinary system. Okay, so now we're gonna take a look at the kidney. We're gonna cut the kidney up, and then we're gonna see what's inside the kidney. All right. Again, you should be drawing this with me, right, to understand more. And so there is the there is our kidney right here. Okay. So this is the ureter. Okay. And this area that's closer to the to where it enters the kidney, that is called the renal pelvis. Okay. Now, excuse me, if we cut the kidney using a coronal plane, a frontal plane, right, so cut it this way, we can see that the inside has a lot of these triangular structures. Okay, if you know what it is, please tell me. What are these triangle things that I'm drawing? This looks like a slice of orange or something like this, slice of lemon maybe. Okay, what are these triangles? You know what? Let me just do a poll just to see. Have you guys watched the recordings yet? I know it has been a busy week with all the tests and stuff. If you've watched it, please choose A. If you have not, choose B. No judgment here. I just need to know. I mean, if most of you haven't watched it, I'm not going to keep on asking questions. Okay, fair enough. So that's going to be the renal pelvis. And these little triangle stuff, that's called the renal pyramid. Okay, renal pyramid. And so you have renal pyramid here, renal pyramid, and then, there are maybe like, 7 to 10 of these. I'm just drawing 3 to keep it simple. Okay. Now, our kidney is surrounded by a thin fibrous casing. And let me try to do this. So there's going to be a case that surrounds it. Like that. It's a little bit tough, a little bit stretchy, elastic. If you ever go to the supermarket, you might see like, pig kidneys or cow kidneys that you can buy. You can poke them and try to feel what it feels like. It's pretty much very similar to the human ones. And this is called a renal capsule. Okay, fibrous casing that goes around the entire kidney. Okay. Now, so earlier I mentioned the outer part of the kidney is called the renal cortex. Okay, the outside of a structure of an organ is often called the cortex. Remember the cerebral cortex, for example. Okay, so there's the renal cortex right here, cortex here. And then we can see the renal pyramid here, renal pyramid, and the space between here, right, where there's no pyramid, that is called the renal column. Renal column. Okay. And if you take these two things together, okay, the renal pyramid plus the renal column, that equals to the renal medulla. Okay. So the cortex is the outside of the organ, and the medulla is the interior of the organ. And what's important to know is that the renal medulla has higher osmolarity. Okay, while the cortex has lower osmolarity. Okay, it's saltier on the inside than it is outside. And that is important in terms of, you know, how the kidney works. We'll talk more about it later. Now, at the tip of our pyramid, we have these, you know, little knobs at the end. Okay? If you think of this as a slice of watermelon, okay, the tip is like the sweet part, you know, that you bite off. That's this tip right here. Okay? That tip is called the renal papilla. Renal papilla. Does anybody have any questions so far about, you know, what I'm drawing? What is it that we're looking at here? Okay. So earlier we talked about the nephron, right? Remember this guy? The ball, and then a U shape and comes back out. This is a nephron, right? So where are the nephrons? Well, the nephrons are all over the place here. And they are microscopic. So like you basically won't be able to see it if you open up kidney. You need a microscope for that. But you know, I'm just going to draw one here to help you visualize. Okay, this is not to scale, right? So there's going to be the Bowman's capsule, proximal tubule, loop of Henle comes back up. It just goes into the medulla and then comes back up. Distal tubule, and then you're going to have the collecting duct that goes all the way down. And I'm going to use a different color to show you another nephron, perhaps. Okay, you might have another one here, right? Bowman's capsule, proximal tubule, loop of Henle, distal tubule, and collecting duct. Okay, and you might have another one here. Okay, all the collecting ducts will eventually feed towards the renal papilla. Okay. From the renal papilla, where is it going to go? Well, it's going to go into an empty cavity, sort of. That's immediately outside. Okay. It's a little bit hard to show here, so I'm just going to color it in for you. So this part here, that's immediately outside the papilla. That is what we call the minor calyx. Okay, I'm just going to put it right here. Minor calyx. Okay. And then outside of the minor calyx, you've guessed it, it is called the major calyx. Okay, small space followed by big space. That's basically what it means. Okay, right here. Major calyx. All right, so I'm going to zoom out. For those of you who want to like take a picture or screen grab it, You can do that, and then I will explain more. Okay, let me know when you guys are good, and I'll proceed. Now, in your course pack, study guide, there is a diagram, right? That looks like this. Right, this one. I don't know if you can see. Oh, you cannot see it. I turn on the sorry, I turn on the virtual background. You cannot see, but I guess I can go here. There is a diagram right here, right? And you can label it yourself. Okay, I basically just went through the whole thing with you. Okay, so this is of course done in the recordings. So if you need someone to guide you through it, you can watch my recordings, or you can try to label that yourself right now, since we just did it, right? Are we okay? Are we together, guys? Any questions so far? Okay. So the urine, right? The urine. Inside the nephron, we have the filtrate, and the filtrate is going to become the urine. And it's going to go down the collecting duct. And from the collecting duct, it goes to the renal papilla. From the renal papilla, it drips out from the papilla into the minor calyx, then to the major calyx, and then out the renal pelvis into the ureter. That's basically the path of the urine flow. And that's what we need for this particular question, right? Okay, so that was the reason I went through all that stuff. Okay, so let's start with the collecting duct right here. Right, collecting duct is the last part of your nephron. And remember, you have thousands of these nephrons filtering your blood at a rate of like 1.2 liters per minute. That is a very, very large amount, large volume of blood to filter. So from the collecting duct, we will go into, once again, the renal papilla. Okay, the little knob at the tip of the watermelon, right? And from there, it goes into the major calyx. Sorry, minor calyx. Small one first. Followed by the major calyx. Okay. Then where does it go? Anybody remember? Looking at a diagram, where do we go after the major calyx? Anyone? Where do we go after the major calyx? What does your diagram say? That's what I'm gonna ask you, right, on the quiz. You know, where is it gonna go, right? And the correct answer, yep, that is the renal pelvis. Okay. From the renal pelvis, it's gonna go into the ureter. Following the ureter, it will go into the urinary bladder. Urinary bladder. And then into the urethra. Urethra, and then out of the body. And that is the journey of a drop of urine. And go through all these things before it comes out. All right, next part. List the blood vessels a drop of blood would pass through, starting at the renal artery and then ending at the renal vein. There's a bunch of stuff that it's going to go through. Okay. Maybe let's draw it as well. Okay. Why don't we draw it as well? Okay. So that you will have it's better than looking at a diagram. Okay? When you construct it yourself, I think you will have a better chance of understanding it and remembering it. Okay. So here we go. Okay. A new kidney. Okay. Please draw a new kidney. There's our new kidney. Okay. I'm going to draw one pyramid here. And I'm going to draw another one. Oh, sorry. One pyramid, two pyramid, and what the hell? Let's draw another one. Three pyramids. All right. So here we will have the renal artery. Sorry, just gotta make more space. Now, as soon as the renal artery comes in, it is going to split into smaller ones. Okay, it's going to split into smaller ones. These one, the smaller ones, they are called segmental. Segmental artery. Okay. So smaller branches. After that, the segmental artery will split and it will go up the sides of the pyramid, like that. Okay, it's going to go up the side of the pyramid. So here is another one that will go up the side of the pyramid. Go up the side of the pyramid. Okay. So these ones that are going up the sides, we call them interlobar artery. Okay, interlobar artery. Then they will curve around the outer edge. Like that, it will curve around the outer edge. And same thing for this one, right? Like I'm just not drawing it, right? It curve around the outer edge and forms what we call the arcuate artery. Okay, it forms an arch. That's basically what it means. And from the arcuate artery, we're going to have a bunch of smaller ones that are going to be coming out like this, like little hairs that are coming out. Okay. All these small ones that are coming out into the cortex, we call them cortical radial artery. All right. Questions? All right. So I'm just going to take out a slice so that we can draw it a little bit better here. Give it a little bit more detail. Okay, so this is the renal pyramid, right? Just going to write down. Renal pyramid. We have the interlobar on the side. Need a thicker pen. There's the interlobar on the side, right? Coming up from the segmental, interlobar, and then the arcuate, right? On the outside. And then we have all these smaller ones that are coming up, okay? So I'm gonna zoom in on one of these things. I'm gonna make it extra big. Okay, so this one, I'm gonna label it for you, the CRA, right? Cortical radial artery, the CRA here. That is going to branch off again into these sideways ones. Okay, we're almost there, I promise. Okay. So these sideways ones, they are called afferent arterial. All right. Afferent arterial. And then the afferent arteriole is going to turn into a web of capillaries. Okay, kind of like a ball like that. And this ball is called the glomerulus. Glomerulus. Okay? Capillary. It's capillary. And it's very leaky. So what happens is, and this is it, okay? When the blood goes through the kidney, remember you have a lot of these vessels. When the blood goes through the glomerulus, because of the leaky nature of the glomerulus and the high pressure in the glomerulus, contents of the blood will get squeezed out. Okay, so you might have water coming out, you will have glucose coming out, a bunch of stuff comes out, okay, except for big things like blood cells and proteins, they don't come out, but all the the fluid component, right, a lot of them come out, and stuff that are dissolved in the fluid, including the nutrients that we talked about, the waste products, all that stuff is going to come out, and we are going to capture it. Okay, so remember our nephron, right, the structural unit of the kidney. There's the ball, right? So the ball is the Bowman's capsule. Bowman's capsule. I'm not spelling it right. Bowman capsule. Okay, so the Bowman's capsule is going to capture all the thing that comes out, right? And the stuff that comes out becomes the filtrate. And then it's going to go on to the proximal tubule, right? The loop of Henle, and so on and so forth, right? Okay, so all this stuff that comes out will pass through the nephron. Okay. And along the way, you're going to adjust the content of the filtrate. Okay, but this is the collecting duct, right? And remember, after the collecting duct, it's going to go to the renal papilla, so on and so forth, right? The stuff that we wrote earlier. Right? So that's the relationship between the blood vessels and the nephron. Each of these afferent arteriole is going to open up into a glomerulus, and then it's going to feed into a Bowman's capsule. Okay, so you have like thousands of these things, right? In total, feeding into numerous nephron, and all of these are going to be working together to produce the urine. Okay, so I hope that shows you the relationship between the blood vessels and the kidney structures. Again, I'm going to take a moment and pause and see if you have any questions. I can hear you screaming through the computer. It's like, no more, too much. Now, I have some good news for you, okay? Historically speaking, and I've been teaching this course for like, I don't know, like 12 years or something like that now. So historically speaking, quiz number 3, which is the one on the kidney, is very poorly done. I don't know why, okay? Like whether it's in person or virtually online, it's very poorly done. So I am going to give you 2 chances for the quiz instead. Okay, so you can do it 2 times if you want. Okay, and it's going to be the average of your 2 attempts. Okay, so let's say you do it once, you get, I don't know, 60 or something, and then you do it again and you get 80 the second time. Then the final score will be 70. Okay, so you will have 2 chances to do it for quiz number 3. So hopefully that will help you a little bit. Now, try to take advantage of it. Like if I were you, what I would do is, maybe take the first attempt in the morning and then see how you do. Right? If you get an 85 or something, maybe don't bother doing a second attempt. But if you didn't do so well, maybe you get like a low 70 or like high 60 or something, do not immediately try the second attempt, okay? Like study a little bit first, right? You have seen the questions. You have an idea of what kind of questions there are. Maybe go study a little bit and then try again at night, Okay, so what happens in before is the people, I don't know what they are planning to do, but then I can see when you actually take the quiz, right? So people actually do it, 10 p.m. at night. Okay. And then as soon as they finish the first one, they don't like the grades, and then they try again right away. That's not gonna help, right? If you do the same quiz twice without studying in between, you are probably going to either get the same mark or like a worse mark, right? Okay. So in order to take full advantage of these 2 attempts, I suggest you do it once in the morning, and then if you don't do well, study a little bit more and do it again at night. Okay. So hopefully that will help. Okay. But seriously, no questions here. Everything makes sense. You totally understand this. All right, tough crowd. But I'm gonna assume that you're okay then. Now, from the glomerulus, okay, not all of the blood is gonna come out, right? Like I said, you're still gonna have some water in the blood. Large things like proteins and cells, they don't come out. So all that stuff will leave the glomerulus. Oops, wrong color. All that stuff will leave the glomerulus. And this vessel is going to be called the efferent arteriole. Okay. And this actually will open up into a web, okay, a web of capillaries and other network that will be adjacent next to the nephron at all times. Okay, so this network is called the peritubular capillary. Okay. So after the peritubular capillary, right, we're gonna change color now. It is going to become it's actually gonna go all the way back here and become the cortical radial veins. Okay. And then it's just backwards. From the cortical radial veins, you're gonna have the arcuate veins that going back down. Okay. And for some reason, we don't actually have the segmental artery. It would just go out and become the renal veins. Okay, and back to the heart. So that completes the entire blood circulation. All right. Now here is a professional drawing from your textbook, or from like, you don't have a textbook, I forgot. This is just a diagram I got from an older textbook. And here you can see all the blood vessels, right? So for each of the red one, you're gonna have a corresponding blue one. Cortical radial veins are next to the cortical radial arteries. Interlobar arteries are next to the interlobar veins. And like I said, there are segmental arteries, but then there are no segmental veins. Okay. And this is the relationship between the blood vessels and the nephron. Right. So, this is simplified diagram. So you see the renal artery coming in, and then we're skipping all the details that we talked about. Eventually, it will become the afferent arteriole, opening up into the glomerulus. Okay, so everything that's leaking out of the glomerulus will be captured by the Bowman's capsule, and then it's going to continue as the filtrate within the nephron. Eventually, it's going to go to the collecting duct. Okay, but along the way, right, you can see another network of capillaries. Okay, so from the glomerulus, it becomes the efferent arteriole, which then opens up into these peritubular capillaries that I was talking about. And eventually, the peritubular capillaries will go back out, and it would exit the kidney and merge with the renal veins. Okay, so I hope that makes sense. Now, after explaining all of that, we can finally go back to here. List the blood vessels that the blood will go through. So starting at the renal artery, we are going to go into the segmental, right? Segmental artery. I'm just going to put A. And then after the segmental artery, we're going to go up the sides of the pyramid. That would be the interlobar artery. And then once we get up to the edge of the pyramid, we're going to turn around and round the outer edge and form the arcuate artery. Okay. We're going to have many, many small ones that are coming off from the arcuate. Remember, like all these little hairy ones. Right? Those are the cortical radii artery. Okay, the CRA. The text people, right? CRA. And then from that, we're going to have like little ones that are branching off. That would be the afferent, AFF, afferent arterial, which then opens up into a network of capillaries, which we call the glomerulus. From the glomerulus, the blood, whatever is not filtered, will enter the efferent arterial, okay, which then opens up into the peritubular capillaries. And then from that is just backwards. Okay, so we have the cortical radii vein. Okay, so this time it's CRV, like the car. And then from that, we just go backwards, right? The arcuate vein, interlobar vein. Okay, and we skip the segmental vein, and it just goes directly into the renal vein. And then from the renal vein is going to go into the inferior vena cava. Okay, back to the heart after that. That's it. Okay, urine formation now. In urine formation, there are three major steps. And we have filtration, which is kind of like a crude process that takes out a lot of the stuff in the blood. Okay, you might accidentally take out, actually you will take out valuable stuff, nutrients as well that you want to take back later on. So that occurs in the absorption step. And in the secretion step, you will basically get rid of stuff that was missed in the first step. Okay, so the filtration process is going to involve the glomerulus. Okay, glomerulus plus the Bowman's capsule. Okay, and in the filtration process, the movement of materials is from the blood in the glomerulus into the filtrate. So whatever is filtered out of the blood, it enters the Bowman's capsule, and as soon as it's in the Bowman's capsule, in the nephron, we call it filtrate. Okay? Filtrate is the stuff that will become the urine later on, okay? Absorption, you're gonna take back things that you want to keep, okay? Like glucose, you want to keep. Amino acids, you might want to keep. Right? All those goodies. Right? And you need to take back a lot of the water as well. Otherwise, we get dehydrated quite quickly. So absorption is going to occur at the proximal tubule. It's going to happen in the loop of Henle, distal tubule, as well as collecting duct. Okay, so along the rest of the way, you're going to be taking back stuff from the filtrate back to the blood. Okay. So filtrate is the stuff inside here, and then we're gonna be absorbing back into the peritubular capillary, which is the web that opens up around the entire nephron, right? Secretion is to take the stuff that you missed from the blood. Maybe there is some more waste product that you missed in the filtration step and you want to get rid of it. And that's going to be taking place in the process of secretion. And it will happen at the proximal tubule, not the loop of Henle, but the distal tubule and the collecting duct. And the stuff is going to be coming from the blood in the peritubular capillary. Okay, so from blood to filtrate. All right. That is that. So let's go through this in more detail by filling in this chart. Okay. So first we have the first step here. Right? What is happening at the renal corpuscle? Right? This was mentioned in the lecture. Renal corpuscle is basically the glomerulus plus the Bowman's capsule. Okay. Remember, afferent arteriole, glomerulus, efferent arteriole, right? And then you put the Bowman's capsule around it. So this whole thing, the Bowman's capsule with the glomerulus together, we call it the renal corpuscle. Right? It's like sausage plus the bun gives you the hot dog. And this is the thing that's together, renal corpuscle. All right, so what's happening in the renal corpuscle? Well, I told you a couple of times already, the blood's going to be coming in from the afferent arteriole, and the glomerulus will, it's very leaky, so all the stuff is going to leak out, right? Okay, so at this point, right, glomerular filtration occurs. This process, this filtration process, is relatively non-selective. Okay, relatively non-selective. And it's driven primarily by blood pressure. Okay, the higher the pressure, the faster the filtration rate. Okay, but if someone suffers from chronic high blood pressure, is actually going to damage the nephron. Okay, it's going to damage the kidney, so it's not a good thing. Okay, but within normal range, higher pressure means higher filtration rate. The walls of the glomerulus and the walls of these capillary structures are porous. Okay, which means leaky. So if you have high pressure squeezing fluid through a leaky wall, stuff are going to be coming out, right? So all that stuff that comes out will be filtered into the Bowman's capsule. Okay. So what remains in the blood? Things like protein. Proteins are very big. They won't go through. And blood cells. Right? We don't have, we're not supposed to have blood in our urine, right? So the white cells, the platelets, the red blood cells, they remain within the blood, while things like water, not all of it though, right? Like, a percentage of the water, things like sodium, glucose, right? All these things are filtered into the Bowman's capsule. Okay, once it goes in the Bowman's capsule, we call them filtrate, right? Filtrate. Filtrate is what will become the urine, as I mentioned multiple times already. So this process, right? The result of this process creates a protein-free plasma. Okay, protein-free plasma. Remember, if you take blood and you spin it down, you will have the hematocrit at the bottom, and you will have the plasma at the top, right? This is the plasma. We learned that a long time ago. Now the plasma has a lot of stuff in it, right? It's mostly water. There's going to be the electrolytes, waste products, and proteins. So if you take away the protein, then you get a protein-free plasma, which is what you will get after filtering it at the glomerulus. Okay, so this is the first step of urine production, the filtration process. It's relatively non-selective. You end up losing a lot of good stuff that you want to keep for the body, but that's okay. It is what it is. Then we're going to move on to the next part, the proximal tubule. Right, here is the Bowman's capsule again. Right, I'm just going to draw the whole nephron in case you not following. Bowman's capsule, we've done that now. So we're talking about the proximal tubule, which is this part. Okay. So at the proximal tubule, absorption takes place. Absorption. Sometimes it's called reabsorption, like you're taking it back. Doesn't really matter. Absorption takes place. Okay. We want to take back some important stuff. Okay. So at the proximal tubule, 60 to 70% of what you just filter out in the previous step will be taken back to the blood. Okay? And this is a process that involves both passive and active transport. Okay, some of it will just happen down its concentration gradient. Others you need to have a pump or something. And it is tightly regulated by hormones. Okay. So under the effects of hormones, you might take back more or less of certain stuff. The amount of substances that can be taken back depends on the number of carriers available. So each substance has what we call a threshold level. Threshold level. Okay, so for example, this is the proximal tubule. Okay, proximal tubule. And on it, we're going to have some transporter for glucose, let's say. Okay, transport glucose transporter. Okay. And next to the proximal tubule, we will have our peritubular capillaries, right? We talked about this earlier. Hope you remember. Okay, so let's say the filtrate is coming by now. The filtrate has water, electrolytes, so on and so forth, and then it's going to have some glucose in it. Okay, so we want to take back these glucose. Okay, so the transporter will pump the glucose back into the blood. Okay, pump it back into the blood. So that's what happens normally. Right? Now, suppose the person has a lot of blood sugar. Okay, maybe the person is diabetic or something. And lots of sugar. So if all of the protein pumps are working at full speed, okay, they're going as fast as possible at maximum capacity, then you won't be able to absorb all the glucose. Some of the glucose will pass through. Okay, so once all the carriers or the transporter for a particular substance has maxed out its capacity, you will no longer be able to absorb any more of that substance. And that's the threshold level for that substance. Okay, one way to think about this as is you think of people working on a conveyor belt. Okay, so let's say we have some people here, okay, at a conveyor belt, and then their job is to let's say these are, I don't know what they are, like potatoes or something. Okay, let's say their job is to pick out the bad potatoes. Okay, bad potatoes. Okay. This is a conveyor belt. If all of them are working as fast as possible, and then there are so many bad potatoes to pick, eventually they will not be able to catch all of them, right? Okay, it will just pass through. So these people are kind of like the transporter. Okay, and the bad potatoes are like the glucose, right? If they are working at full capacity, then they have reached their threshold level and you won't be able to absorb them anymore. So they will end up in your urine. Okay, I don't know if you heard this before, if you know this, but people who have diabetes, right, their sugar level in the urine, it could be elevated, right, if they're not on any medication. Okay, last point here. Blood pH is controlled by secretion of hydrogen ions and absorption of bicarbonate ions. Right, so maybe that deserves some explanation right here. Okay. We produce carbon dioxide as a part of cellular respiration. Okay, so all the carbon dioxide is exposed to the water in our body. And this is going to create, anybody know what this is going to create? Like from a chemistry class, for example, carbon dioxide plus water, what is it going to be? Anyone know? You guys taking chemistry, right? What is that going to be? Really? Nobody knows? Okay. Carbon dioxide plus water. Oh, thank you, Jyoti. It is going to be H2CO3, carbonic acid. Okay. Now, H2CO3 is actually going to break down into H plus and HCO3 minus. And now, in chemistry class, they might tell you reaction goes forward, but actually they can actually go backwards as well. They can shift back and forth. All right. So H plus is what will contribute to, so high H plus equals to low pH, right? So how can you control blood pH? Well, if you have too much H plus in the body, then you can get rid of some of it by secreting it into the urine. Okay, if you have too much H plus in the blood, let's just put it in the urine, and then you can pee it out, and that could help, bring your pH back up a little bit. In addition, in addition to removing the excess H plus, you can absorb bicarbonate from the filtrate. So when you absorb bicarbonate in the filtrate, the extra bicarbonate that you've absorbed will mop up the excess H plus. Okay, so these two can combine and it will form H2CO3. Okay? So by getting rid of excess H plus, either by secreting it or by, absorbing more HCO3 so that you can combine with the extra H plus, both of these mechanisms will help control the blood pH. Okay. All right, let's take a quick break, 5 minutes, and then we will resume in a bit. Okay, let's keep going. Next, we have the descending limb of the loop of Henle. Right, so as you go down, as the filtrate goes down the descending limb, what happens is you will be absorbing water from it. Okay, so this section of the nephron is impermeable to solutes. Right? It's impermeable to Na plus and Cl minus. And as filtrate moves along the descending limb, water is absorbed through osmosis. Okay, and the concentration of the filtrate will increase. Okay? So if I draw the loop of Henle here, okay, water will get reabsorbed into the peritubular capillaries. Okay, and what's drawing the water out is the increasingly salty environment. Okay, remember earlier today I said that the medulla has higher osmolarity than the cortex, and so that increasing osmolarity will help draw the water out from the filtrate. Okay, so the filtrate that goes down, as it goes down, the concentration will go up. Okay, so I'm going to use a darker color to show that. Okay, so like that. And the concentration will be the highest at the bottom. Okay, will be the highest at the bottom. So highest osmolarity. Okay. The next part is going to be the ascending limb. Okay, so ascending limb is opposite. Okay, instead of absorbing the water, we are going to be absorbing Na and Cl. Okay, and this is absorbed by active transport. Active transport. Okay, you're going to be pumping it in, and as you remove the NaCl, the concentration of filtrate will decrease once again. And so just go back to this picture in a second. You know, as you go up, as the filtrate ascends, you know, it's going to get less and less concentrated. Okay, like that. So if I ask you on the test, on the quiz, where is the osmolarity the highest in the loop, in the nephron, it will be the bottom of the loop of Henle. Okay. Finally, we are at the distal tubule and the collecting duct, and normally they are impermeable to solutes, and adjustment to the contents of the filtrate can only be made by hormones. Okay. And the amount of substance absorbed or sometimes secreted is controlled by various hormones, such as in the lecture we talk about several. There's aldosterone, and then there is things like antidiuretic hormone, ADH. And there's also one that we mentioned is ANH, atrial natriuretic hormone. Okay. We sort of covered this already, but we can talk about it again. Write the chemical equation for the bicarbonate buffer system in the blood. Okay, so we have the H2CO3. It's an acid, so we put it's dissolved in water, so we put aqueous, and this could go, you know, left or right. When it goes to the right, the bicarbonate will dissociate into hydrogen ion plus bicarbonate ion. Okay. So a buffer system is a system that will resist small changes in pH. So the blood pH is 7.35. If it gets too basic, then you will shift to the right and make more H+, right? And if it's too acidic, then it will shift to the left to remove the excess H+. So that's basically how it works. So explain how the buffer system resists changes in pH. Okay, so when pH increases, okay, pH increase means it's more basic. That means it's not enough H+, right? The buffer reaction shifts right to produce more H+. Okay. So you're replenishing what you don't have enough of. On the other hand, when pH decreases, okay, too much H+, the buffer reaction shifts left to remove excess H plus. Okay. Explain how the kidney plays a role in maintaining the buffer system. We talked about it earlier, right? Add the proximal tubule. You can secrete. H plus can be secreted. And bicarbonate can be absorbed to help increase, to help, increase blood pH. Okay, so if your blood is too acidic, then this is going to be happening. If it's not too acidic, then this is going to be happening less or not happening at all. Okay, moving on. I actually explained this already in the lecture recording, but given that not many of you have seen it, maybe I'll go through it again. And so there is something called the RAS, which is the renin angiotensin aldosterone system. Purpose. The purpose of the RAS is to increase blood pressure. Okay. So here we go. Trigger. There is a drop in blood pressure. Why? Can someone tell me why you might experience a drop in blood pressure? Like an otherwise healthy individual, why would the person experience drop in blood pressure all of a sudden? Dehydration, that's right. What's another reason? Or what's another reason why you have drop in blood pressure? No? Okay. Excessive blood loss, right? Excessive blood loss. When you change position, you might feel a little bit lightheaded if you just get up really quickly because the blood gets pulled down. But generally, blood pressure will not change drastically when you change your position. But good try though. Okay, so this is a drop in blood pressure, right? And these are some possible reasons of why you might have a drop in blood pressure. Okay, so let's take a look at this. We have the afferent arteriole. And by now you should know what I'm drawing. Okay, what is that thing again? Just checking to see if you are all with me. What is this little mumbo jumbo thing looks like? What is it called? That's a glomerulus, right? Okay. Now, located on the walls of the afferent arteriole is a special cluster of cells. Okay? We call these cells JGA. I'll put the name here in case you it's in the notes anyways. Is a juxta. Juxta means right next to. Juxtaglomerular apparatus. Okay, so it's a group of cells that's next to the glomerulus. That's what it is. So the JGA will monitor the blood pressure as the blood goes through the afferent arteriole. Okay, and what happens is, in response to a drop in blood pressure, so low BP, the JGA will release an enzyme. Okay, the name of the enzyme is renin. It is an enzyme. It's not a hormone. The difference between an enzyme and a hormone is that the enzyme catalyzes a reaction, hormone signals another part of the body to do something else. Okay, there's no signaling involved in the enzyme. Okay. Now, your liver, which is this thing, it's on the right side of your upper abdomen, right? The liver is going to make a lot of proteins. We talk about how it makes albumin, makes things like fibrinogen. And one of the things that is related to what we're talking about is that it makes something called angiotensinogen. Angiotensinogen. And this is inactive, and it's circulating in your blood all the time. Right? Angiotensinogen. Ogen is inactive, right? We talked about that before. And in the presence of renin, angiotensinogen will get converted into angiotensin. Oh my goodness. Angiotensin number 1. Okay. Angiotensin number 1. Angiotensin number 1 will get converted into angiotensin number 2 by something called ACE, okay, which is angiotensin converting enzyme. And this is from the lungs. Okay, so the lungs makes the ACE. Okay, so if your blood pressure is fine, then everything stays as angiotensinogen. But if it drops, then the JGA will release the enzyme renin. Renin will convert angiotensin into number 1, and then ACE from your lungs, right? ACE will convert number 1 into number 2. Okay. Now here is a question that you might see on the, oops, gotta erase all the answers here. Okay, here's a question you might see on the, well, there are more questions. There's a lot of questions actually. Let me erase them all. I wish they have a button that would let me just erase everything. Unfortunately, they don't. Okay, so here's a question you might see on the test. If the level of renin in the blood is high and the level of ACE is low, which of the following is correct? Okay, so let's think about what we wrote down earlier. Angiotensinogen is going to get converted into angio number 1 and then angiotensin number 2, right? From angiotensinogen into angiotensin 1, we need renin. And from 1 to 2, we need ACE. So the question here says, if renin is high and ACE is low, which of the following is correct? Okay, so let's see. If renin is high, that means we will be able to convert angiotensinogen into angiotensin number 1. Okay, but if we don't have enough ACE, then number 1 will not be able to get converted into number 2, right? So if this is the situation, we would have low angiotensinogen, right? High angiotensin 1, and then low angiotensin 2. Right? Basically, we have a bottleneck here, or we cannot go forward because we don't have enough ACE, right? So the correct answer is this one. Okay, low angiotensinogen because all of it get converted into number 1, but then we're stuck at number 1. Okay, nothing gets only a little bit get converted forward, right? Because we have low ACE. Does that make sense? So of course, this question can have many variations, right? You should think about what happens if it's like high, high, or low, or low, high. And then depending on this, the answers will change. Okay. All right, let's go back to here. So what happens to angiotensin number 2? Well, angiotensin number 2 is actually going to go to your adrenal gland. Remember adrenal gland, right? This is your kidney. Adrenal gland is the thing that sits on top of it, right? So the adrenal gland is going to, in response to angiotensin number 2, it is going to secrete aldosterone. Okay, it's going to secrete aldosterone. And another thing that will happen is, in addition to going to the adrenal gland, the angiotensin number 2 will also cause vasoconstriction. Okay, so the vasoconstriction is going to basically decrease diameter, and then it will increase blood pressure, right? Which fixes our problem, right? Our initial problem is we have low blood pressure. So that's going to be, counteracting that. What would aldosterone do? Aldosterone will go to the distal tubule and the collecting duct, and it will increase sodium absorption. Okay, it will take the sodium from the filtrate back into the blood. And then, because you have increased sodium absorption, water will follow. By osmosis. Okay. And that will increase blood volume, which in turn increase blood pressure. Okay. So that's basically how the RAS is going to work. Right? The RAS system is going to help increase the blood pressure. Any question about RAS before I talk about the counteracting force of this? Okay. So, of course, the opposite can happen. Right? Instead of having low blood pressure, we can have high blood pressure. I don't know. Let's put it here. Okay, you can have high blood pressure. Okay, for various reasons. So, if the blood pressure is too high, you do not want the RAS to be happening. Okay, so when the blood pressure is too high, the heart is going to be secreting something called atrial natriuretic hormone. Natriuretic hormone. Okay, so it's a hormone that's secreted from the atrium. Natri is sodium, basically. Okay, it's the Latin name for sodium, natriureum, and basically the hormone will cause sodium to be in the urine. Okay, so the ANH will do a couple of things. Number one, it will suppress renin. So if you suppress renin, then none of the stuff is going to happen here, right? Okay. It is also going to suppress aldosterone. Okay. RAS is only one of the ways that you produce aldosterone. There is another pathway that we'll talk about later that secretes aldosterone. And so, you know, even if you suppress renin, there might still be some aldosterone. So that's why ANH will also inhibit that. Okay. And ANH will also go to the kidney, go to the distal tubule and collecting duct, and it will basically do the opposite. Okay. It will increase sodium secretion. So H2O will be secreted as well. And because you're losing water, you will decrease blood volume, which then decrease blood pressure. Okay, so these two systems, the ANH and the renin, and the RAS rather, they are opposing each other at all times. Okay. So let's put it in a diagram here. Okay. I guess we can do it here. We have some space. Say we have normal blood pressure, normal BP. So that's good. You have your 120/80. And then for some reason, your blood pressure goes up. Okay. Increase blood pressure. Okay. So how do you bring it back down? Okay. Which one do we need to bring it back down? Well, we just talked about it. You need the ANH. Okay. And then the ANH will bring it back down. Okay, by doing all these things. Okay, increasing sodium secretion, inhibiting the aldosterone, inhibiting the renin. Okay. And then if the blood pressure is too low, if you drop in blood pressure, okay, then you will activate the RAS. Okay, which ultimately is gonna cause you to produce aldosterone, right? And then it's gonna bring it back up. So ANH and aldosterone, these are antagonistic hormones. Okay, we'll be learning 3 pairs of antagonistic hormone in the course. This is the first pair. They have opposite effects. And by having opposite effects, they are able to maintain homeostasis for blood pressure. Okay. Now, the ANH has limitation. Okay. Sometimes, if even when the ANH is working at maximum capacity, you are not able to bring the blood pressure down to a acceptable level. Okay, then you have hypertension. So you will need to have medications. There are 2 types of medications that could help fix this problem. Okay, you can take what's called ACE inhibitor. Okay, so ACE inhibitors are drugs that you can take that will literally does what it says. It will block ACE, and if you block ACE, then you cannot convert angiotensin 1 into 2. Then that means you will not activate RAS, right? Okay. So that's a good thing because RAS will push blood pressure up higher, right? And then you can also take diuretics. Diuretics helps you go to the bathroom and like pee more. And by increasing the urine output, you lower blood volume, which lowers your blood pressure. All right. Does anyone have any question? Yeah, I have a private question. So that means ANH increases high blood pressure. No, ANH is released when there is high blood pressure. And then it will do all these things to bring it back down. Okay, to bring it back down. There's a hormone chart for aldosterone. There's something called antidiuretic hormone, which helps you conserve water, and then ANH. The chart is filled in the lecture recordings, so I'll let you watch that on your own. Okay, I'll post the answer for the fill in the blanks. Let's do some post questions. Okay, let's take up some of these questions. Okay, I will launch the poll. Go ahead, the first one. Okay, so looks like you guys are not really participating. So I guess I will just I'll just take them up. It's probably easier that way. Well, I do have 5 answers, so let's share the results. Which of the following is not normally found in the filtrate? Remember, things are too big, don't get filtered out. So proteins will not be there. All the blood cells will not be there. So the correct answer is this one. Okay, E. Okay, next one. The efferent material takes blood to the. So what you want to do for this question is look at what we filled out here. And you can see the efferent material will be taking blood to the peritubular capillaries. So that is the answer. Okay, you just have to look it up. This one we just talked about. Okay, why don't I let you try that? The 5 of you who are participating anyways. Okay, see what you have. Okay, just look at what we drew here. This chart here. Okay. ANH. Look at all the things that it does, and then choose the answer. All right, I have 2 answers. Waiting for more. Okay, anyone else? Last call before I take it up. Okay. Well, let's see. Okay, ANH will. You know what? Let me copy this question here. And we're going to go back to this chart. Okay, put it right here. ANH will increase aldosterone level in the blood. Well, let's take a look. Okay? You can see here ANH inhibits aldosterone, right? So it doesn't actually change the level of aldosterone. It just blocks them. So that's not the answer. Increase blood pressure. If you follow the pink arrow, all these things happen, which ultimately decreases blood pressure. So that's not true. Decrease sodium level in urine. Well, it says increase sodium secretion, right? Secretion means it goes into the urine. So that's not true. Increase sodium level in the blood. Well, if you are secreting it, then you are taking it from the blood and putting it in the urine. Okay. Look here, right? Secretion, we defined that earlier. Secretion means from blood to filtrate. Okay. So that means if you're putting it in the urine, you are actually not increasing it in the blood. So the answer is increase urine volume. You're peeing it out. Right? Hope that makes sense. Okay, this is another good one. These are all real questions that you will see on the quiz, that you're likely to see on the quiz. So take a look at this one. Okay. Which of the following would initiate the osmoregulatory adjustment? Osmoregulatory is just like a fancy word for adjusting, sodium, right? Through the renin, angiotensin, aldosterone system through the RAS. Okay. So when do we need RAS? Let's go over here and see. RAS, which begins with the secretion of renin, is triggered by low blood pressure. Why do you have low blood pressure? When you have low blood, you would have low blood pressure, for example, if you have dehydration or excessive blood loss. Okay, so not having enough fluid in the body, or if you're losing a lot of blood, will drop your blood pressure, which would then start the entire RAS system. So here it says, which of the following is going to trigger RAS? Okay, 3 out of 4 of you chose drink several glasses of water. Does drinking a lot of water cause your blood pressure to drop? Is that dehydration? It's not, right? That is the opposite of dehydration, if anything. So that is not the answer. Okay. Shuffling the driveway for several hours on a cold winter days. We all experienced this in the past winter, probably, right? Lots of snow. Is that going to cause you to have dehydration? Is that going to cause you to have blood loss? Okay. Probably not, right? So that's not the answer. Like you might sweat a little bit, but you're not going to be like dehydrated. Mowing the lawn for several hours on a hot summer day. I say that's going to make you sweat a lot, right? That's going to make you have a bit of a dehydration, right? Unless you rehydrate yourself. So this will drop BP because of dehydration, right? Okay, and that is going to be the answer. Right? Eating a pizza, sure, you are eating some sodium, but you are not going to be as dehydrated as mowing a lawn for several hours in a hot summer day. Right? Sleeping will definitely not be an issue either. So the answer is C. Okay, so whoever you are, the only person who got it right, good job. Let's do another one here. I'll do this one with you. Damage to the renal medulla will interfere with the function of, Okay, so remember this is the pyramid. Okay, and then you have your nephron, which looks like this. So if you damage the medulla, which is all this part here, right, we are gonna affect the function of, well, let's see what's in the medulla. Right? The loop of Henle is gonna be a problem because it's in the medulla, right? Collecting duct is gonna be in the medulla. Okay, everything else should be fine because all these stuff are in the cortex, right? Okay, so no problem with these structures. Okay, well, this one is a mouthful. Let's do it together. How do ADH, okay, ADH, I didn't really talk about it in the tutorial. ADH basically helps conserve water. Conserve water, okay? When you are severely dehydrated, you will have ADH that helps you reduce urine output. Okay. RAS, we know, is going to increase the BP. Right. So how do they work together to maintain blood osmolarity? Let's see the first one. ADH monitors appropriate osmolarity by reabsorption of water, and RAS maintains osmolarity by stimulating sodium reabsorption. Okay, so this is true, right? RAS will help with sodium reabsorption. We talked about that already. Okay, and ADH will also reabsorb water. Okay, I should probably write down for you, but again, it's in the recording. Okay, ADH absorb water, absorb H2O at distal tubule plus collecting duct. Okay, so this is true. ADH and RAS work antagonistically. ADH stimulates water reabsorption during dehydration. RAS removal of water when it's excess in the body. That's not true. Okay, RAS does not remove water. Okay, this is not true. RAS absorb water, right? ADH will be released, and RAS will be activated when a person is severely dehydrated. So when you are dehydrated, that means you will have low BP. Okay, so ADH will be released to absorb the water, and RAS will be activated because of that reason. So that is correct. Only when they are together at the proximal tubule will reabsorption of essential nutrients back into blood take place. That's not true. Okay, they are independent of each other. The mechanism is completely different. Both stimulate the adrenal gland. That's not true. ADH does not have anything to do with adrenal gland. Only the RAS does. Okay, so the first one and the third one are the correct answers. Last one. Diuretic, I briefly mentioned about that. It's a drug that's sometimes used to treat individuals with hypertension. The drug inhibits the reabsorption of sodium by the loop of Henle. Remember the loop of Henle? On the way down, you're going to be absorbing water, right? H2O. So if you have a diuretic, it's going to block this. Okay. So what effect would this have on the osmolarity in the nephron? If you are not reabsorbing the water, then the filtrate, remember earlier I said the filtrate would be the most concentrated at the bottom because you're absorbing all these water. But if you're blocking it with a drug, then it won't be as concentrated as before, right? Okay, so here, osmolarity would be higher than normal in the collecting duct. That's not true. Okay, if anything, it should be lower, right? So here it says osmolarity would be higher. So anything that's just higher, it's not true. Okay. Osmolarity would be lower than normal in the proximal tubule. Proximal tubule comes before the loop of Henle, and the drug is acting on the loop of Henle, right? So anything before that point will not be affected. Okay, so the correct answer is osmolarity would be lower than normal in the collecting duct, okay, which is right here, right? Because you did not absorb all those water, so now it's going to be less concentrated. Okay. Like I said, this is a very involved lecture, a lot of concepts. Watch the recordings if you haven't already. Study hard. And like I said, the quiz will be, you will have 2 attempts. But unlike the lab quiz, right, it's not going to be the best of the 2 attempts. It's going to be an average. Okay, so you don't want to just like, go in blind and wing the first one. Okay, you do want to study a little bit first. I'm just looking at the calendar right now. The quiz is going to be next Tuesday. Okay, next Tuesday. So you have a week to no, you don't have a week. You have 1, 2, 3, 4, yeah, 5 days. Okay. If you have questions, of course, or need a little bit of extra help, feel free to email me. I'll be available throughout the weekend as well if you need help. Another thing I should mention is this. Let me just show you the calendar. Next week, it's going to be a long lecture. Okay. Now, I don't know if you probably don't remember because at the beginning of the pandemic, we only had 13 weeks as opposed to 14 weeks. Okay, but now we're back to the 14-week schedule. So what happens is I only have recordings for the first part of lecture 10, which is the bone parts. Okay, and now that we are back to 14-week schedule, we have more time. We actually have to learn the muscle as well. I don't have a recording for the muscle part, so instead I'm just gonna teach it during the tutorial. Okay, so next week we're probably gonna likely go to the entire 3 hours. Okay, so do plan for that. Okay, but the quiz is going to be next Tuesday, and I'll post the information about it later on. All right. Yeah, thanks for coming, and good luck on the quiz. I'll see you next week.